Hello, my name is Joshua Sudduth. Today I will be speaking about mechanisms for marine bioluminescence. Marine bioluminescence is defined as the light emitted by organisms in both the land and in the sea. It is expressed in over 76% of deep sea animals, and over 97% of cnidarians exhibit bioluminescence. This includes corals, jellyfish, and sea anemones. Also, 90% of fish also display bioluminescence. This shows how important bioluminescence is as an ecological trait. Marine animals use bioluminescence in the predator-prey relationship, where the predator, like the deep-sea anglerfish, will attract its prey with the bioluminescence on its head, and the prey will defend themselves from predators. For example, some jellyfish shoot out bioluminescence to distract predators. Same with the vampire squid, which shoots out mucus that is bioluminescent, which distracts the predator and the prey can get away. We can use bioluminescence as genetic reporters. They are able to deliver 10 to 1,000 times higher assay sensitivity than fluorescent assays. And they use the firefly luciferase as the intracellular probe. Chemical excitation opposed to photon excitation in fluorescent assays almost completely erases the assay's background. No photons are introduced like fluorescent assays, which likely completely erases the assay background. It can also be used to study gene expression, protein-protein interaction, and disease progression. For example, biosensors use genetically engineered cells to generate bioluminescence in response to a target analyte. So how does bioluminescence work? The first article I will be discussing talks about the integration host factor and how it synergistically binds to DNA with LuxR to co-activate quorum sensing genes in Vibrio harvae. It is written by Ryan Chaparian et al. So this picture here at the top goes a little over how quorum sensing works. So the first part here, we see a little bacteria cell with some red dots. These red dots are autoinducers. As we have more cells in a confined area, we also have more autoinducers. When there's enough autoinducers, it will bind to the LuxR, as shown in the third picture, and will work to activate the bioluminescent genes. So as we increase in bacterial cell density, we will see an increase in bioluminescence. And that is how quorum sensing works. The integration host factor is a nucleoid-associated protein which works in chromosome condensing, or in this case, gene expression. The integration host factor binds to multiple sites in the LuxCDABE promoter and bends the DNA in vitro. It works with LuxR to activate the LuxCDABE bioluminescence genes. It can bend the DNA up to 180 degrees, which is important for promoter regulation. IHF can be split into two subunits, IHF alpha and IHF beta. The LuxR is a transcription factor and is responsible for precise timing and expression levels of bioluminescence. In this study, the author wants to learn how IHF and LuxR work synergistically to bind to control timing and expression of bioluminescence. To test this, they create new strains of Vibrio harvae with one of the strains have the IHFA and IHFB encoding genes removed, so IHF cannot bind to the DNA. And they also created a strain where the LuxR encoding genes were removed. They compared both these strains to the wild type by measuring relative light units over OD600, which is the cell density. Here we see the results for the experiment. On the bottom x-axis, we see OD600, which gives a reading from a spectrophotometer at 600 nanometer wavelength, indicating cell density. And the y-axis shows bioluminescence measured in relative light units, or RLU for short. The wild type gene is shown in circles, and we see bioluminescence becomes expressed at around 0.1 to 0.2 cell density. And for the variant strains, where IHFA and IHFB were removed, as shown as squares, and where the LuxR encoding gene was removed, as shown in the triangles, we notice that at around 1 or 2 cell density, that is when bioluminescence becomes expressed in the organism. What was found is that IHF controls the timing of bioluminescence, just like the LuxR transcription factor. And as predicted, both IHF and LuxR work together to activate LuxC-DABE gene expression. The mutants having a 10-fold difference from the wild type strain indicates that IHF binding is required for full transcription activation of the Lux genes. This diagram models the quorum sensing control of the gene, where the LuxR transcription factors are correlated to cell density, and the IHF is binding to the DNA to bend it and further control gene expression. From the results, we gathered that bioluminescence is shown to be under quorum sensing control, and that IHF and LuxR synergistically bind to DNA for LuxC-DABE gene expression. The second article I will be reviewing is titled Bacterial Bioluminescence, Light Emission in Photobacterium Phosphorium is not under quorum sensing control. It's written by Lisa Tennant et al. The methods in this article were similar to the previous article, in which cell density was measured with a spectrophotometer at 600 nanometer wavelength, and light emission was measured in relative light units with a luminometer luminoscent ascent. Growth of P. phosphorium was done on an ONR7A modified medium, which contained minerals and salts. It was then transferred to a 100 milliliter flask containing 50 milliliters of ONR7A modified medium, 19 degrees Celsius, with shaking done at 150 revolutions per minute. Growth was measured for 35 hours, with sampling done at hours 4, 9, 15, 22, and 27. Since little was known about this mechanism, a quantitative reverse transcription PCR LuxHerb gene analysis was done at all 5 sampling times to determine mRNA expression. The results of the experiment are displayed here, where on the left we see a bigger graph inside of a smaller graph. The smaller graph represents your normal quorum sensing bacterium, while the bigger graph represents P. phosphorium. P. phosphorium differs from your average quorum sensing bacterium because as cell density increases, so does the relative light units, which are indicators for bioluminescence. Where in quorum sensing bacterium, the bioluminescence gene is not expressed until there is enough autoinducers to create the LuxR transcription factor. The figure on the right shows what genes are being expressed during all 5 sampling times, which differ from your typical quorum sensing bacteria. Quorum sensing bacteria are indicated by the LuxR transcription regulator, which is not present in P. phosphorium. And to conclude this article, it should be known that QS is not always present in bioluminescent bacteria, and the idea that quorum sensing is directly associated to bioluminescence should be avoided. It was also shown that bioluminescence is not always regulated by transcriptomics, which is evident in P. phosphorium. This figure on the right shows how your typical quorum sensing bacteria have the LuxR quorum sensing transcription factors, and on the bottom, P. phosphorium does not contain the same transcription regulators as quorum sensing bacteria. Overall, we see that somewhere between 76% and over 90% of marine organisms express bioluminescence. And we see how bioluminescence can teach us about human diseases and give 1,000-fold higher sensitivity than fluorescent assays. And that there are many more mechanisms present that are not quorum sensing, and that bioluminescence can be expressed in more mechanics possible than we thought, and there's plenty more to learn about when it comes to bioluminescence. Here are a few of my references. Please let me know if you have any questions.